Thank you. I would like to start by thanking the organizers, not just for inviting me, which is very nice, but for putting such an impressive meeting together. It's been great so far. Right, I'm going to talk about why glycans matter. You probably know what proteins are and why they matter, but what about glycans? Some people would probably have made the connection already. Glyco is a word that we borrowed from the Greek language, glucose, which means sweet. Most people would associate sweetness with sugar, and in this context is perfectly correct. But sugar can actually come in many forms, and none of them are sweet. Table sugar is just crystallized sucrose, which is made of glucose, which we got here, and fructose, which is there. Sugars are also known by the technical name carbohydrates because they contain well, carbon. We got hydrogen there, and we got oxygen there. But it comes in some other forms. Cellulose in wood is just a bunch of glucose stabilized by hydrogen bonds. And I hope you will agree with me that it doesn't taste sweet at all. So these are free or ligand sugars, but it is in antibodies that we're going to see sugars that are actually linked to a protein, in which is called protein glycosylation. These glycans are branched carbohydrates permanently tied to a protein molecule, which is what I'm showing here as a semi-transparent surface. Mind you, protein glycosylation makes a significant yet fairly small part of a glycoprotein, which is what we call glycosylated proteins. As you can see here, this fragment of an antibody is basically bigger than a few nanometers. And there are quite a lot of non-sugar atoms inside. This doesn't prevent the glycans from often driving the function of the protein they're attached to. And another thing that is very important to protein glycosylation, we need to keep in mind that while genes provide blueprints for building glycoproteins, glycans are actually attached by the cellular machinery. So there is no blueprint here. This means that, for example, human and insect cells would produce different glycosylation profiles, which is a nightmare. And talking about nightmares, this is a model of SARS-CoV-2, the causative agent of the pandemic. The elongated proteins coming off its surface are spike glycoproteins, which is what the DNA and mRNA vaccines are instructing our bodies to produce. And because we're humans, our cells will attach human glycans to those spikes, just like in a real infection, which is cool. Glycans can have many different kinds of sugars, so it's useful to have a cartoon diagram like this, which has the spheres and cubes representing individual sugars. So the blue cube is glucnag, the green sphere is mannose, and so on. These will help us understand what's going on in simulations. This is actually a very long simulation done by one of my collaborators, Elisa Prada. And we can see how one of the glycans twists and turns to accommodate itself in a pocket in the receptor binding domain, which is, of course, key for infection. So as we can see, aside from somewhat shielding the spike glycoprotein from antibodies, glycans are also involved in the function of the system. So it's great that we can simulate the mechanics of molecules and that this can aid our understanding of their behavior, but these techniques are reliant on the accuracy of the atomic models that they are setting in motion. A simulation of a wrong atomic model will produce misleading results. So we need to make sure that all the atomic models are as correct as possible. So how can we make sure that we're producing correct atomic models? I think at this point, we need to see how we determine atomic models of glycoproteins in the first place. These are representative maps from the two main techniques, so X-ray crystallography and electron cryo microscopy, and some of the resolutions they may achieve. Obviously, this is outdated but for cryo-EM because they're doing better now. This is from 2020. So worse resolution means more guesswork, obviously, and in the absence of expertise, it can lead to modeling errors. Also, working with sugars is not easy. they can interconvert between these 5 forms, which is a mixture of open chain form in the middle, 2 ring forms, pyranose and furanose rings, and 2 anomeric forms, alpha and beta. And each one of them should have a specific 3-letter code in the protein data bank. That's hard to remember. To make things worse, they may adopt different ring conformations and different anomeric forms, like here at the reducing end. These may coexist in a crystal structure. Pyranose rings can take a number of shapes, ring puckers, ring conformations, depending on how you want to refer to them. And these depend on how the atoms are arranged with respect to each other. Here we have glucose, and if we try to get a better view, we can get rid of the hydrogens. Things look a bit more clear, and if we hide everything but the ring, the conformation becomes fairly obvious. It is a chair, and sugars like chairs. So of all the different conformations that sugars can adopt, the chair is the most comfortable one for them because it minimizes the clashes between the things that are attached to his ring, the substituents. Yeah, good. Obviously, simulations can show how conformational transitions would work in case an enzyme is distorting a sugar. As you can see in these simulations, in the middle is the chair conformation, which is symbolized by a C, letter C, and the energy barrier that needs to be cleared in order to go to other conformations is quite significant. Therefore, I think it is fair to assume that normal sugars will always be in a chair conformation, except in the case of external intervention by an enzyme, for instance. However, we have shown that these realities are not well reflected in structures of glycoproteins. As we can see from this conformation analysis of all the glycoprotein sugars available in the protein data bank, there are a lot of distorted sugars in the middle of this plot. So boats, skew boats, and so on. These models, basically these ones on the left, are the result of modeling mistakes and refinement mistakes and interpretation errors that have highlighted the need for new carbohydrate-specific methods that will allow us to produce models like the ones on the right. Misleading models may lead to misleading mechanistic interpretations, which in turn will mislead others. So I think as a community, we should strive to avoid them. And this is what we've been doing to help. We've got this privateer software, which will scan your structure for unlikely ring conformations, biology-defying glycan compositions, poorly fitting models, and will help you improve them in combination with standard model refinement and building tools such as CUD, RefMag, and others. It can also help with dictionaries, and this has allowed us to produce new conformational restraints for pyranose sugars that Rovi has introduced, and it will also model complete glycans in sensible conformations, which can be used in molecular mechanics and so on. Like in this case, where we've got a grafted glycan. The privateer software, it has been around for like 6 years now. It's been useful in a lot of situations. It is available for both CCP4 and CCPM, and the newest MKIV release will bring all the AlphaFold related functions, plus a Python interface that would allow us to deploy interactive interfaces for model building programs such as CUD and Chimera X. One of the latest developments that we've introduced allows users to check the glycan structure and composition against Glycomics databases, which are mirrored offline. This allows people to spot inconsistent linkages, such as these core-linked fucose, which is linked in beta 6. The software can suggest alternative compositions, as in this case, and also can help you get a more complete structure, like I'm showing here. This is how we run Privateer MK4 from CCP4i2. This is already released. You will need a model and reflections. You got a number of settings you can set. You can choose your expression system, configure the vector-based plots, and ask it to compute permutations in case you got the composition wrong. You run it, it's all parallel and super quick. So well, it won't take very long to run. It offers a number of plots, conformational plots, real space versus B factor. In this case, it has identified that there is a sugar that is distorted, the conformation is distorted. But for instance, if we look at the glycan plots, we can see that they have identifiers for both Glycogen and Glyconics databases. Although this sugar is in a higher energy conformation, it doesn't really affect the structure of the glycan. In this other case, we've got lots of distorted sugars that are marked as red excess. And if we look at the plots, it will suggest some alternative structures. To try and reduce the chances of producing distorted models, and as Robbie has said already, we have generated new restraint dictionaries for pyranose sugars using a combination of H-drug and privateer. This incorporates a set of validated unimodal torsion restraints, which we have tested extensively, and can be incorporated in refinement, just like any other sort of restraints. So we've tested this to make sure that the torsions work properly. And interesting, with future versions of RefMag, you'll be able to activate the torsion restraints separately from the rest of the sets. Also, the torsion restraints will match the low energy conformer that is in the dictionary, which is what you place in the model building program. Robin has already talked about this. We've managed to reduce the conformation outliers significantly using the unimodal torsion restraints. And also, if you look at the different ligands, it doesn't really affect when they have a well-determined high energy conformation. Another interesting thing is that they can reduce our gap. So in terms of overfitting, it will reduce that. In the process of doing this, we've also got in touch with developers and make sure that we made sure that we obtained consistent results with different software. So for instance, Pyrogen, H-drug, Grade, Phoenix Elbow, and so on, as you can see here on the right. The MKIV version of Privateer, which will come as an update to CCP4, will also bring support for getting tryptophan mannosylation right. The mannose residue was nearly always found distorted in the project data bank, as shown here. That was probably not helped by the diagram in our reference textbook, The Essentials of Glycobiology, which showed a beta link instead of the expected alpha 1. This has been updated for the forthcoming 4th edition, and it will show the latest structure information, which shows a mannose in 1C4 inverted chair linked to in an alpha linkage to the tryptophan. Now, the new Privateer will help you detect problems with tryptophan mannosylation and fix them using 2 different mechanisms, one with external torsion restraints, the other one with the standard dictionaries. The new Privateer MK5 will also incorporate distortional analysis, so you can check your linkages in glycosylation and compare to what the rest of the structures look like. And finally, just to finish this, we've also introduced new functionality that allows us to graft molecular dynamics equilibrated glycans onto models, including AlphaFold ones, where we generally see that surface complementarity is somehow retained, as in this potential CH5 interaction between the tryptophan and the apolar phase of the glucanac. The structures come from a growing library of glycan models produced by our collaborator Elisa Fada in Ireland. These are conformational representatives from very long simulations. We've got so all these high mannose glycans, and now we're working on complex glycans and some all glycans. So those are coming very soon. We're also doing some machine learning on the PDB and PDB redo sites. As Robbie has said, PDB redo contains improved information. So we're actively using that too to try and predict protein glycosylation based on sequence and interactions, such as the ones depicted here, CH bite interactions and hydrogen bonds.
It's 15 minutes now.
Yep. And
Thanks.
with this,
Perfect.
I would like to conclude. This is my team plus Jordan. I didn't have a picture of Jordan. He's been working on torsional preferences, and collaborators, obviously, Robbie and Rob. We've done lots of things together. Elisa Pada from Ireland, and then in Joshi, Elena Segirodaki from Oxford. We're working on tryptophan mannosylation, and then Martin, Frank, Frederic, Melanie, Samir, and Herad, and CCP4 and CCPM. And thank you very much.
Thank you. We are probably kind of fighting with time, so if you don't feel offended, could you answer the Slack questions?
No problem.
Am I right that there is nothing in the Q&A now? Yeah.
There's
okay.
nothing in the Q&A.
So, yeah, maybe we can give a new second chance to Jane.